The Moss Report is brought to you by Mycolife, makers of the world's best mushroom extracts, including the Moss Method mushroom formula, available exclusively at mycolife.us. There are a number of scientific terms used throughout this podcast. When you see this icon in the top left corner, that means we've included the term and its definition in our glossary. Link in the description. Hello, healers. My name is Ben, and this is the Moss Report. I'm here today with my father, author and founder of the Moss Report, Dr. Ralph W. Moss. And we're joined with our very special guest, Dr. Matthew Halpert of Immunocine, which is both a clinic and a protocol, if I understand correctly. Gentlemen, thank you both very much for being here, and we're looking forward to this conversation. Matt, if I can, I just want to prompt you to give us a little bit of a background of your own personal journey in terms of how you've arrived at both your protocol and in the clinic that you have now, in terms of your education and how it led you to where you are.
Sure. No, thank you. Thank you both for having me and for letting me discuss this. I'm happy to cover all these things. So my background, I have a doctorate in immunology. I'm an immunologist. That is what I desired to be. And once I earned that degree, I spent roughly 10 years as an academic researcher in the Texas Medical Center, which of course is a very robust area for research. Predominantly working on, the immune system, but specifically dendritic cells. And so the reason, and a lot of what I say, of course, is not me. It's me and my group, my colleagues, my research people. It's no science is an island, right? And so working with this group, we were really focused on dendritic cells and, more importantly, why they seem to be so utterly worthless in the clinic. Dendritic cells were discovered in the 1970s. Of course, by Dr. Ralph Steinman, and no one really debates their importance in biology. No one really challenges that they are very, very critical and necessary to an optimal, complete immune response. That's their job. Their job is to see a threat and then organize a response to it. And when you are sick or chronically sick, right, something has gone wrong. There's a problem there. Admittedly, when I started doing this research, my focus was not cancer necessarily. It was more on the side of the immune system and dendritic cells. And as we move forward through time and we start learning different things, of course, I've come to view cancer as much as an immunological disorder as anything else. Absolutely, there are mutations and toxins and environmental cues, all those things that trigger a cancer. But cancers are always kind of being triggered. Sometimes cells replicate more than they should. And we have cells specifically capable of seeing that and eliminating it before it's a problem. Meaning if it is a problem, something's gone wrong. I mean, that's just, level set right there. Something's not working right. And part of our research was, and some of it's just curiosity, and we can talk about the actual findings, but really long story short, dogma, dendritic cell dogma is, not entirely accurate. And that of course happens. That's how science moves. The thinking has been that dendritic cells blindly will consume or absorb a threat, bacteria, virus, whatever, and communicate that to the rest of the immune system. And they have little recognition inside themselves as to what that threat is. Patently false. And we have very, very strongly shown this through a variety of efforts and data. I mean, it's all published, we're not really hiding it. As it turns out, the dendritic cell in the inside, intracellularly, has a fail-safe mechanism. When you want to launch a nuclear missile, you have to turn both keys at the same time. That makes sense. You don't want to accidentally blow everything up. And so the dendritic cell has something similar. When it experiences a specific type of threat, and I'll try and not get too into the weeds here at the beginning, but when it sees this threat over in this pathway, the thought was, there it is, and I will respond. Nope. What it first does is it looks to see if it sees the same pathway over here. Yes or no? And if the answer is yes, then that right there, then and there, changes everything about the dendritic cell. It now, without any other cells or any other signals, has decided I will need to change my entire genetic programming. There is a very real threat. We must activate this full, Th1 immune response. And that is actually as close as we've been able to see. I'm speaking as an immunologist, to a step one in what, people look at Th1 immunity and they look at things like IL-12 and interferon gamma. You know what? Those are downstream of this. We've conclusively shown that this goes first. And so as we kind of had that aha moment in the lab, me and my partners, there were moments, honestly, where you back off and you kind of almost cry, because it is critically important. It does change the way we view dendritic cells and the immune system, which means, now we have to protect it. Now we actually have to push it forward. We really can't. I can't take this other job. How could I be the guy that leaves this phenomenal finding to go do this other thing, right? And so pushing forward, all we did is listen to the skeptics. They would tell us all the reasons we were wrong, because if we were right, we should be able to do this, 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 and this. Those things were the basis of all the publications. I'm like, yeah, you're right, we should, and we did. And you fast forward now, and this science itself is now the platform of two different companies, one US-based as a more traditional biotech that has a variety of FDA clinical trials and has been fast-tracked by the FDA a couple times, is working, I would say, steamrolling towards an approval. We can talk about some of that. But then, because it is just biology, and there are cancer patients today that don't have 10 years to wait for all these approvals, in no way do we want to denigrate the importance of the FDA or the trials or the statistics. But, if my mom gets cancer and I know that this immunology can help her, I don't really care about the statistics. I don't really care about if a certain checkbox has been checked. And so trying to walk this line where we're certainly not trying to skirt anything, but we did get some protocols approved and launched Immunocine, effectively operating as the closest I can come to it is a compassionate use offering. We're certainly not a walk-in clinic. We do require medical reviews, and we don't really make it that easy to come to us. But if this science can legitimately help a patient, either on its own or as an adjunct to their treatment, it just almost seemed like a moral obligation to find a way to get it out there ethically. So happy to talk more about any of those things, but you opened the door and I talked for 10 minutes. So.
No, that's fine. I enjoyed hearing that story. And before I encourage my dad to weigh in with his set of questions, I would like to sort of back up to something that you said about the effectiveness in clinics as of present. And my understanding is that dendritic cell therapy and the way it's used in immunotherapy protocols is more like a targeted delivery system for things like the Newcastle virus and so forth that use the dendritic cells' ability to permeate membrane and travel in the bloodstream to deliver essentially a payload into a tumor that then raises its profile or changes the nature of the tumor microenvironment in such a way that's similar to the way the immune checkpoint inhibitor drugs function. But from what I'm hearing from you, it sounds as though your approach is very different from that. Is that a fair statement?
That would be a fair statement. We're not using dendritic cells as a delivery vehicle to the tumor per se. So, right, there's only one FDA-approved dendritic cell treatment, right? Provenge for prostate cancer. It's based on science from the 90s at the latest. It doesn't really work. That's just, I don't really think that's a secret. But so the purpose of, oncolytic viruses in general, right, or viruses, the type of immune response needed to eliminate a virus is a Th1 immune response. It is. Our body can see a bacteria and physically get rid of that bacteria. It actually cannot get rid of a virus. That's not how it works. It doesn't say, there's a virus, I'm going to blow you up. the way it works is the virus infects a cell, and of course it's going to replicate and eventually lyse, and our immune system, our body, what it will do is actually identify the infected cells, inducing them to kill themselves before that rupture, and you do this until there's no more infected cells and the virus is then gone. That's exactly what you want in cancer. You want those cells to be eliminated, right? And so the merits of an oncolytic virus are strong. I've been a big fan. They haven't really worked. There's some caveats and flaws, but one of the goals is if you get a virus into the cancer, let's say every cancer cell in your body and only those cells become virally infected. Well, maybe your immune system doesn't see the cancer, but it sees the viral infection, and same thing happens. It eliminates the cells. You get a lot of those same signals, those danger signals. It all works together because it is the immune response you want. Now, if the virus is not specific to the cancer or doesn't infect all the cancer, and remember, your immune system is being targeted and educated against the virus, not the cancer. So there are some fallbacks there, but obviously there can be benefit. You can get a lot of good out of that. What we're doing is putting the cancer information into the pathways of the dendritic cells, so the dendritic cell sees the cancer now how it would see the virus. We're not flooding the cancer with the virus. We are using the cancer as the target of this antiviral mechanism, if that makes sense. And so what the dendritic cell learns, because the dendritic cell would see a viral infection this way and this way. And that's all we're doing with the cancer. It's a targeting platform. So no, we're not using the dendritic cell to deliver anything to the tumor. That wouldn't actually help. We're using the dendritic cell to go to the nearby lymph nodes to educate the rest of the T cells, killer T cells, the natural killer cells. Wake up. Like, you should have seen this. We all should have seen this a long time ago. We see it now. We're a little behind, but that doesn't mean the fight's over. For most patients at this point, you'll get lymph node swelling, you'll get fatigue, you'll get low-grade fever. It's an immune response. Their body finally realizes, oh, we're sick. And now those immune cells will flood around the body looking for anything with those signals that it just saw in A and B there.
And this was the protocol that you basically discovered while you were at Baylor, is that right?
So there's really, and I gotta be careful here, right, who I leave out, but there's really a triumvirate of scientists involved. Dr. William Decker is my partner at Baylor College of Medicine, made the very, very initial discovery. And so he, playing around in a petri dish, basically said, hey, what if I add 1 and 1, does that 2, or do we get more than that? And it turns out you get 9,000. What happened? Something drastic happened. And of course, and so this was back in right around 2010, right? And he's being a curious scientist. He's following it and he's sticking with it, even though people are telling him, this makes no sense. You're not right. Your stuff's contaminated. That's what it's got to be. I joined him kind of shortly after that. And we talk about this finding. There's no importance yet. We don't know what this means. Could be important. I think it's fascinating. I love that all the older scientists are saying, you can't be right. It can't be right. If it's right, we have to rewrite the textbooks. I love that. So I said, yeah, let's do it. And then I go on to confirm, and then drastically start adding. Because one of the tricks, of course, one of the little secrets in academic science is you spend your whole life or education becoming this phenomenal scientist in the lab, only to reach a point where then you have to sit in an office and write grants all day, and you don't actually do the science anymore. So I became kind of the hands on running all the experiments. And then shortly after that, joining me was Dr. Vanaja Kanduri, who also is still at Baylor. And together, the three of us really continued to develop, understand the mechanism. We know exactly what machinery is doing this. We know how fine-tuned this is. We know it's at the level of the amino acid overlap, bound to the MHC molecules. We took it through a variety of cancer models. We have pushed it forward into clinical trials. So it's really, and then of course there's been a variety of other researchers in the lab, but I would say that the three of us. So I'm not going to go on here and say, yes, it's all me. It's not. And Dr. Decker deserves all the credit for making the very initial discovery. But I think the three of us share it pretty evenly, and we're good with that. Now, I am the one admittedly, that, saw some opportunity to help people today that just don't have time. And so I kind of went out on my own and started researching, can we license this to a tourism group? Like, well, who can do this now? And found a bunch of nobody, and ultimately did it myself around joining up with another scientist, Dr. Rafael Gonzalez, who I met in Cancun, who happened to be running an ISO 7 certified lab operating in the space of autologous cell therapy. And I said, well, if you guys can do what you're doing, you can do this. And Immunocine was sort of born around the existence of that lab and the scientists. There were actually no doctors when this started. We can get doctors. The science has to be done correctly. That was the key thing for me. And so we sort of bore Immunocine out initially just to be present as a word of mouth sort of opportunity. And word got out, and we've been growing pretty quickly. So, I go back to, it does reshape the way we look at dendritic cells and the immune system. It does. People did not know dendritic cells are aware or comparing what they're absorbing and communicating, and they are. And if there's a defect there, I actually have a hypothesis that itself may lead to a lot of immune dysregulation and even cancer. I mean, that may be in and of itself a problem for some patients. But for a lot of patients, we can take this to the lab and we can fix it. It's as simple as that. And it doesn't guarantee a cure. All I tell people really is, you're trying to beat cancer, you're running this race, you're running as fast as you can, you're doing everything you can, this supplement, this protocol, this. As an immunologist, until you get a physiological immune response engaged, which is what we do, I would debate you're not even on the racetrack. You're running really fast, but the racetrack is that way. And how about if we can help you, if we can put you on the racetrack, I think it gives us better shots at outcomes. And we can talk about some of the stats and things, but did you?
Well, now I don't want to interrupt you because I'm enjoying hearing everything that you're saying. And I have a lot of questions bubbling in my head now, but I'm going to turn it over to my dad because I know he's got some questions for you that might probe a little bit more deeply than even the late questions that I might ask. So, Dad, whenever you're ready, I think Matt's gearing up to do the deep dive with you.
I will talk as much as you guys want, clearly.
What was that discovery? Where the 1 + 1 = 99 or whatever.
Yeah, and I'm happy to talk about it. We obviously have patents and we've obviously published it, so it's not like it's not out there. Basically, there need to be overlapping antigens simultaneously bound to MHC class 1 and MHC class 2. There is a gigantic complex inside the cell that continuously scans the class 1 and class 2 molecules. And when it sees basically a perfect match, that's what activates. That's what will cause a conformational change in the equipment. You'll get the release of some strong Th1 promoters that, as it turns out, kind of launch a Th1 response, which leads to genetic changes in the dendritic cell. A lot of genetic changes it's not one thing. The whole thing changes. And I'm simplifying a little. We call it double loading. You have to double load. You have to get it in both. But if you don't double load correctly, it doesn't work. It doesn't recognize it. If you're off even just a bit to the dendritic cell, those are not the same, and it doesn't activate. So it's both very simple, which almost all biology is when you understand it, and at the same time, it is meticulous and complex. I just got off a call with our manufacturing regulatory team involved in all the clinical trials. I mean, the regulatory system, there's just, I don't know, it's thousands of pages and documents and this failure and this, and there's a lot that goes into it. But at the core of the biology, it is that simple. And again, we've published repeatedly on it in Blood and Science Translational Medicine and FASIV. I'm very proud of my FASIV paper. It's very nerdy. You'd have to really want to dig into the weeds, but it's all out there. And it very clearly shows that if you double load and you mess the double load up by even 2 amino acids, it doesn't work. If you restore the overload, it comes right back. I mean, it is that fine-tuned. It is so fine-tuned that you can play with it at this little degree based on what's bound to the MHC molecules, and it either is on or it's off. Biology does not work that's not a fluke. Like clearly that is a purposeful scanning mechanism. And with what we see afterwards, and I can tell you about some of the things we're seeing in the trials or with the cases. We're moving needles we're moving needles in glioblastoma, for example, that have quite frankly never been moved. And we're just getting started. So people will get 3 shots of dendritic cells. Why 3? Why not 5? We got to start somewhere. I don't know. Like maybe it probably can be improved. It probably can be optimized. But even with what we have, we're moving the needle pretty drastically. So again, it's not that dendritic cells, yes, historically dendritic cells have not worked very well in the clinic. I would argue to you and to your listeners, that's because no one's ever turned them on before. Of course they didn't work. You can be as mad as you want that the car didn't get to 60 miles per hour or didn't make it from Florida to California. You didn't turn it on. Who knows what dendritic cells can do? We're only going to just find out now. And I just made a whole bunch of people angry that work with dendritic cells elsewhere, but I'm telling you, if you look at the science, we're not wrong. And that's what I base everything on. We're not wrong. It doesn't guarantee what the outcome is. It doesn't say, no, you don't need to do other treatment. I'm not saying that, but we're not wrong. This is a fundamental mechanism to getting an immune system turned on and moving down to a very strong Th1 pathway.
Fascinating. You mentioned the Federation of Experimental Biology. There's a big convention every year, and you're presenting there. And what is the reaction that you get when you present this work at FAS? What do you have, posters
No, I mean we've presented at different meetings, and we've gone to AACR and AI and ASCO. So now, to be fair, now we're presenting a lot of clinical data. And it does change. When you have clinical data, when you can say, look at how the patients are doing in this trial, they've exceeded expectations, et cetera, it gets a lot easier. In the earlier days, oh my gosh, no, presentations were met with a lot of scrutiny, for sure. Like, we would get, some of the papers we had trouble publishing, this goes for grants too, because we were told, dendritic cells don't work that way. And it's just, but we're showing you they do. No, they don't. You must be wrong. We're not going to pay attention to this. And no, people on our team, someday, but not today, we're not a big name. We're not necessarily at the biggest institution in the world. Like, that stuff does matter. And so that's not been the story now. We've gotten multiple R1s. It's getting a little easier to publish. All of a sudden, people are coming out of retirement to join this project. We have some Nobel laureates who kind of want to be a part of this. Like, everything's different now, but for a good 7-8 year swing, it was a lot of skepticism, despite what the data show. In more recent times, we are actually now, for the most part, it would seem to be accepted. It's not dogma. We haven't replaced that yet. Nothing's been rewritten in textbooks, but it doesn't seem as crucified as it once was when we talk about this, because again, there is a lot of data. With that said, a lot of people don't really truly understand, dendritic cells, especially if you're not an immunologist. And so some of them, it's exciting, but we may leave and a week later they may see a lysate loaded dendritic cell report and say, how's this different again? Okay, it's very different, right? That's going to be single loaded, not double loaded. So
the double loaded, you're presenting what? mRNA to the cell and proteins? Is that what makes it double loaded?
What we do, for example, right, is we'll take someone's cancer. We don't need a lot. We will isolate the mRNA, the total mRNA. Now, for the record, you can do a lot of things in the lab you can't do in the clinic. We can make, we can grow up cell lines, for example, in the lab that we can have as much as we want. That's not realistic in a clinic. So we actually had to come up with a now patented way to amplify mRNA, because you're not going to get enough from, for example, a pancreatic cancer patient. It degrades too quickly. But we're going to get that mRNA, the thing that encodes the cancer. And we're not cherry picking. We're not picking our 10 favorite antigens. We're not doing that. That's, we don't need to. The dendritic cell will do that. It's designed to do that. We will feed that to the dendritic cell, electroporation or what have you. At the same time, we will then, give it a swath of cancer lysate or protein so that it can put those things on the class 2 pathway at the same time it's putting these things on the class 1. And rising to the top, what will be noticed by the dendritic cells on the inside is the overlap. 99% of a cancer, or maybe more, is healthy, right? I mean, it's not like a cancer cell is 100% mutated. It has a cell wall, right? Cell membrane. That's how much encodes that, right? I mean, most in there is correct. Yet doing this in 20 years in the lab, in clinical trials, in compassion, we have not really been able to turn on off-target effects. I will not tell you it's impossible, but we just really haven't seen it.
Yeah, when you say off-target effects, you mean side effects?
Well, yes and no. I don't mean side effects. I mean, we don't accidentally target, your leg or your liver. Like there doesn't appear to be rampant autoimmunity. So one of the concerns, right, with the checkpoint inhibitors is that long-term use can induce autoimmune conditions. Makes sense. Those drugs, as powerful as they are, and I'm generally a fan in the right context, they're not cancer specific, right? They're going to help immune cells respond stronger, harder, longer when maybe they shouldn't. And around a cancer, that's exactly what you want. But around other places, no, it's bad. It's going to be autoimmune. We just haven't really seen that. We didn't see it in the trials, not in the trials so far. Side effects, we see side effects. Those are there's immune response, swollen lymph nodes, fever, muscle aches. It depends. Honestly, a lot of it depends where the cancer is. If you have cancer in your breast, you're probably going to feel swelling and pressure there as the immune system floods in. If you have it in your prostate, nothing. Fatigue. I mean, you don't really feel your prostate, and those patients have had almost no symptoms. The glioblastoma patients in the clinical trial had no attributable severe adverse events, and that's dealing with the brain. If you have a ton of cancer in your spine, that's probably not going to feel great. We're turning on inflammation in your spine. There could be swelling of these lesions now in your spine. Sometimes for those patients, we require prophylactic radiation, palliative radiation, or we maybe just don't even treat if it looks too dangerous. We don't want to destroy someone's quality of life. And it becomes a very gray area, of course, is what's worth what to who. But so side effects or effects of the immune response are relatively universal and based on tumor burden and tumor location. Off-target effects, which another word for that I think would be incorrect autoimmunity, we haven't really induced, meaning the dendritic cell is seeing the mutated, the neoantigens, the mutated epitopes, and targeting that. And so are there other groups out there that will transcriptionally look at your cancer, say we like these 10, epitopes or neoantigens, we're gonna make peptides? Fine, all that's fine. Are you double loading the dendritic cell with them? Yes or no? And the answer always has been no. Okay, then you're just not going to activate step one. You just have to accept that. It just isn't activating it. Could you design peptides to do both? Yes, and we've done that, and those are in some of the publications. Now, of course, if you do it now, depending on where you are, you're infringing on all our patents, and the Texas Med Center is going to come after you. I wouldn't recommend that. Just for anyone listening, it's not worth it. But again, it's easy and it's not. On the one hand, I don't mean to make it sound like anyone can just come out of grade school and just do this, but on the flip side, it is pretty simple, again, when you understand what you need to have accomplished. You got to get the same stuff overlapping on class 1, class 2, and when you do that, there you go. Kind of that's it.
So I heard a word back there, and the word was electroporation. To me, that would kind of imply that you're, quote unquote, injecting the mRNA and the proteins into the dendritic cell. Is that accurate? In other words, you're presenting it, but in the sense that you're actually physically pushing it into the dendritic cell?
The mRNA needs to get inside the cell. That's where it does its work. That doesn't have to be electroporation, but that's a very common one. You could use a viral vector if you wanted to. You just have to deal with that. The proteins, no. The proteins will be phagocytosed. When a dendritic cell gets virally infected, one of the first things it does for 15, 20 minutes is it goes through this rapid phagocytic burst. Why? It's been noted and no one really knows why. That doesn't make any sense, right? It's infected. It's doing that because it's looking. It recognizes the viral infection on the inside, and it's now eating the outside to say, do I see other viral debris and things out here that are the same? Is this a viral infection? HIV does not infect dendritic cells. Ebola hijacks dendritic cells. It's like a lot of these severe viruses know what's going to happen if they get double loaded. And so to that point, this is not a cancer thing. It's a Th1 targeting platform. Yes, we've done experiments that this could work for HIV, for example. We haven't done enough, but in theory it could. And so that gives us the excitement that we have a trial for glioblastoma, we have a trial for pancreatic ductal adenocarcinoma, we have one coming up for melanoma. It's not cancer specific. We just need to be able to get a sample of the cancer, properly loaded into your dendritic cells, which we can do in the lab, put those cells back near non-compromised draining lymph nodes, and we got a shot. We're going to get your immune system turned on. It's going to become engaged. We can show you that, and we do at Immunocine. We do our own flow cytometry. We have these beautiful immunological panels, which we provide to every patient to show them. We didn't just tell you we gave you dendritic cells. We're showing you the dendritic cells. We have data for that, and we're showing you how your immune system has responded, has educated, has shifted. And for 90 to 95% of the patients, they respond correctly. We could argue that's our objective response rate. The patients will respond. The next question is it in time? Does your body have time and the strength? You're just starting the race now. Can you catch up and win? And for some patients, we would say, probably not. You probably do need to do some chemo or radiation or something else to bring the cancer back it's just too far ahead. And so we routinely will tell patients, if that's what we think is the honest truth, we'll say it's not about just how awesome this is. It's you want to live another 50 years, right? That's what we're going for here. And so, And for the record, I don't care what the age of the patient is. I always say we're going for 50 years. Who knows? But we want this to be a speed bump. And so if that means 3 months of chemo to get things under control, and then we do this, it's just very case by case.
But is it somewhat dependent upon just the number game, just the numbers of lymphocytes that you have that the person has to work with?
I think so, but I think so more in theory as an immunologist. So you have cancer in your body. How many cancer cells you have is a who knows, X. Mm-hmm. And it's growing. That's what cancer does. It's growing. What's the rate of growth? We also don't really know. So X to the Y. Mm-hmm. On the other side is your immune system's ability to eliminate it. Currently at 0. Now for some patients, it is on, and those are the patients that will likely respond well to checkpoint inhibitors. If you can give it a boost, you might have a shot. But most patients, it's just 0. Mm-hmm. We fix that, right? Or that's the goal. The goal is to fix it. I can't stipulate a 100% claim here, but that's our goal, is to fix it using this technology. And now it's Z. So Ben, Ralph, me, I don't know how your immune system compares to mine. How strong is yours? How strong is mine? There has to be a theoretical maximum to how many lymphocytes a body can make at one time based on energy constraints. There just has to be, right? At least that's, and so, I think it's a numbers game in that way. X to the Y versus Z.
Do you use things like Neupogen and Neulasta, drugs that would increase the number of blood cells, red or white?
Yeah. During the first week of treatment with us, whether in the trial or Immunocine, is you get 4 or 5 days of Neupogen to get those white blood cells up, because otherwise we're not going to get very many to make dendritic cells. That would be the first problem, is we won't get very many dendritic cells if we don't do that. Mm-hmm. So yes, that's one thing. And we're constantly sort of looking for potential scientifically vetted add-ons. Again, I'm not going to pretend that this is in and of itself the end all be. It's critically important, but hey, is there more? Is there something else we could do to skew things towards the Th1 immune response or increase the numbers? With that said, we also don't want to throw kitchen sinks at patients who have already especially gone through a lot, especially if we don't have the science to support it.
So we spoke with Jim Allison, who was working out of MD Anderson and who got the Nobel Prize for his discovery of, I can't even pronounce it, ipilimumab.
Yeah.
The trade name is Yervoy.
Right.
And what he told us when we interviewed him for our film Immunotherapy, The Battle Within, and also for our podcast, was essentially that when he was doing his clinical trials, that the patients that were getting the treatment, in the absence of a healthy and diverse gut microbiome, there was actually a negative outcome. In other words, they were left only with the side effects from the drug. And those who had a healthy and diverse gut microbiome, he was saying we are getting cures. So I'm just wondering whether you're doing anything microbiome forward, or getting people tested for that, or having anything to do with nutrition or diet and sort of helping them prep themselves for before coming to your clinic.
Yeah, no, it's a great question. So I can't speak to the data per se, but I totally agree with Dr. Allison, who, for the record, is he's our neighbor. We do know him. He's aware of what we're doing. He's very interested. The gut microbiome, I think, I don't think there's any question about how important it is, gut health in general. There's a variety of things that have been linked to better outcomes for cancer patients. We're focusing here on cancer, but quite frankly, I'm sure this is kind of universal. The healthier your gut, the better you do. Even there's been, some publications in the not too far past where, daily making sure your dietary fibers, at least, I think 20 grams a day, pretty drastically improved outcomes for cancer patients who were doing, immunotherapy. I think there was a really interesting study came out, really focused on beta hydroxybutyrate, which is a ketone, and how it can also synergize with some of the, and most things do look at the checkpoint inhibitors as they did. For the record, Dr. Allison made his discovery back around 1990, took 20 something years to get that across the finish line.
Sure.
Science and medicine move slow. Why is this taking so long? I don't know, because that's just how long everything takes. But so, yes, I am a big fan and a believer that a healthier gut is probably going to be impactful. On that note, to separate divisions, remember, we can do a lot more with Immunocine than we could do with the clinical trials, right? Clinical trials are what they are. You're looking at the baselines of what this does. We actually are we've actually created our own we'll call it a version 1 of a gut health shake, basically, to add some things like beta glucans and dietary fiber and there's some good probiotics too, right? Akkermansia and Bifidobacterium are two of my favorite. Not to sell. I don't want to do that, and Immunocine's not doing that. We're not selling supplements. That's not the goal. But do I think strongly enough, as the founder and scientist right behind this, that a healthier gut would lead to better results for patients? I do. And so our goal is to make this and then give it to the patients. It does not cost them anything. I just think if they take that for a couple months while we're doing this immunotherapy, getting it up and running, I think they will respond better. Do I have definitive proof yet that this formula is the winner? No. But my point in that I'm doing it, that we are doing it, is evidence to, we do strongly believe in that. And of course, you bring up a great question, and I'll just be very transparent with you guys. We're in the middle of trying to figure out how much we want to do, right? The more you try and do, the more it gets tricky. How much can Immunocine do? Are we just giving these double-loaded dendritic cells to get patients' immune systems engaged? Or are we also trying to have a preparatory program that uses supplements and powders? Are we also trying to have an adjunct program? Like how, and so we're still feeling that out ourselves as well, as to what is appropriate, because some things are not going to be appropriate. Our team is so loved and so amazing. Almost every patient, will come and end up, it's great. They love us so much. We're very honest. We're transparent. They trust us. And sometimes that's to their own detriment. They go home. They're, 500 miles away, and they want us to maintain the role as primary care.
Yeah.
We can't do that. It's not ethical. It's not right. It's been 3 months since we've seen you, for example, and you're far away. And so that's been we're working on that, is we don't want to overstep and any ethical or medical boundaries there. So what is the appropriate thing how much can we do or should we do?
We look forward to following your progress over the next few months and years and telling people about it.
Thank you.
Well, gentlemen, thank you both very much. It's been a fascinating conversation. I'm really looking forward to sharing it with our audience. But for now, for the Moss Report, I'm Ben Moss. How are you healing today?